Okay, I think I have to have a recap. Let me have a recap. In today's class, we talked about, this class is all about natural products, chemistry. So when you talk about natural products, you're talking about the chemistry of anything that is made by nature, made by God. Okay? Now, in this context, we are going to limit our discussion to chemistry of plants, animal, and living organisms. So, the compound that can be isolated from this are natural products. Natural products are biological molecule, okay, that we obtain from nature. We can have them to be primary or secondary metabolite. When we talk about primary metabolites, we are talking about those metabolite that is needed for survival. Like energy, you need your carbohydrates, amino acid, you need it for growth, fat and oil, you need it for energy and warmth and stuff like that. So these are natural, these are primary metabolites. Now, these are produced in large quantity because it's needed for survival. Then secondary metabolites are those that are produced in plant in minute quantity. They are needed for health. They are source of medicine, and they can be source of pharmaceutical or phyto or cosmetic agents. So that's why they call them secondary metabolites. They are not needed for survival. So these secondary metabolites are also agents in new drug development. So we call them late compound in drug discovery. So when we talk about your quinine is an active from plant like neem, which is called dongoyaro. The active from this plant was isolated by a chemist and derivatized in the lab to be synthesized in larger quantity to take care of malaria. So the protocol is a long process. We call them biosynthesis, okay? Or bio-guided isolation. When you talk about bio-guided isolation, you're talking about, I want to isolate the most active component in an extract. So I have to know the activity I'm looking for. If I'm looking for activity of anti-malaria, so I'm going to test my crude extract with plasmodium, and anyone that is most active on this plasmodium, I'm going to extract it, then test the final isolate to make sure that it's very active against plasmodium. Then I'll go to lab and either derivatize it to make it much more potent and then scale up to make it available in large quantity. So I said you should be able to define the term natural product. In a simple term, we are talking about the chemical substances that is being derived or obtained or produced by living organisms, okay, that have potential that can be either therapeutic or toxicological. So we call them that are going to have specific pharmacological potential or pharmacological effects. So these are so you should be able to define the term natural product. These natural products, we have different classes. Now, there's difference between natural products and secondary metabolites. So if you ask the classes of natural products, it includes the primary and secondary metabolites. But if the question is, what are different classes of secondary metabolites? You're limiting to the alkaloid, the flavonoid, the steroid, and all those bioactive components of plants, animals, or living organisms. So that's the sex. So you should be able to distinguish between primary and secondary metabolites. What's different between primary and secondary metabolites? With examples, you should be able to answer this question without missing words. Now, I talk about that these natural products can be derivatized in nature. We call it biosynthesis, meaning that I can obtain one natural product from another naturally in the lab. So naturally in nature, meaning that reactions also occur in nature, right? The way we can obtain one compound from another in the lab, I can obtain carboxylic acid. I can obtain ester when my alcohol and carboxylic acid react. The same thing can occur in nature. Reactions can occur in nature. So those reactions that occur in nature are what they call biosynthesis. And because this biosynthesis has to do with phytochemicals, meaning I can get one phytochemical from another, we call it biosynthesis of phytochemicals. We all know about photosynthesis, that everything that occurs in plants originates from photosynthesis. And photosynthesis simply means when CO2, which is a byproduct of respiration by man, when man is going to take in oxygen from plants and release carbon dioxide. This carbon dioxide, please control my class. This carbon dioxide that has been released by man is going to react with water in the atmosphere. By the virtue of sunlight, we are going to produce carbohydrates. Process called photosynthesis. Photo has to do with light. So synthesis by light forms our carbohydrates. Now, this carbohydrate is going to produce pyruvic acid. Pyruvic acid is going to lead to acetic acid. Acetic acid is going to form our fats. Okay, fatty acid, which is called polypeptides. Now, the same carbohydrate is also going to give us a ischemic acid. Ischemic acid is going to form our tannins. From chemic acid, we are going to form our chorismic acid. Now, if what I'm targeting is tannin, so from my photosynthesis carbohydrate, from carbohydrate to shikimic acid, my shikimic acid is going to form at my top. It's something like flavonoid. So from my shikimic acid, I'm going to connect to chorismic acid, which is going to link to my aromatic amino acid, which will form my phenyl paranoids, then flavonoid. Then if what I'm looking for is my alkaloid, so from my aromatic amino acid, I'm going to form my alkaloid. So the essence of this scheme is for you to understand that I can obtain one compound from another. Now, how do I obtain my terpenes? Now, if you look at the arrow, you can understand my terpene. Where is my terpene coming from? From that chorismic acid, okay? I'm going to, from my chorismic acid, look at where the arrow is moving. Arrow is moving from choris to flavonoid. So how do we get what you are looking for? Now, you go back. Where is the arrow that connects to amino acid? From amino acid, I'm going to get my mevalonic acid. All terpenes are product of mevalonic acid. So my terpenes are byproduct of mevalonic acid. So because the next slide, subsequent slide we're going to be looking at is my terpenes. So terpenes and steroid are byproduct of mevalonic acid, which is synthesized from amino acid. So this biosynthesis, what can we get from here? The most important thing we can get from here is I want to get my tannins, I want to get my flavonoid, I want to get my alkaloid. This can actually be gotten specifically from the slide. But whenever you're talking about your terpene, you're going to steroid, you're going to talk about they follow a pathway called mevalonic pathway. how is this natural product isolated? Remember, I said natural products are made up of like every plant. That's why you can hear that these plants, this somebody agbo, they said this agbo is taking care of this, is taking care of this. One agbo can take care of as many ailments as more than 10, 12. Why? Because these herbs are made up of so many phytochemicals. So these phytochemicals, they work synergistically. Okay, they work in unity. At times, one can be highly potent, others can be calming the potency of the highly potent, right? That's why the benefits of natural product over the synthetic or your drugs on store is that one, there are some metabolites in this natural product that are calming down the potency or the side effect of those ones that are highly active. So that's why they work in synergy. So when we have an extract is made up of so many phytochemicals. So for us to get a particular one that might be responsible for the whole activity, we do what we call isolation. But before you do isolation, you're going to do extraction, you're going to do separation before you isolate. Isolation is a very stressful technique that involves so many protocols. Before it was obtained as a compound, it was a long. So in this section, we'll be looking at how do we get an active from the whole phytochemicals obtained in plants. So that's what we're going to be looking at. So the first thing we're going to get is that when you get your plants, like you went to the bush and obtained plant, if you cannot use that plant that day, what you can simply do is to dry. And in drying, it's always advisable for you not to dry under the sun. Why? Especially if you're dealing with sensitive part of plant that are like leaves. When you dry them under the sun is going to take away most of the metabolites, especially the essential components that are sensitive to heat or light. Okay, some of them that are not stable by heat, when you sun dry them, you lose them. So that's why it's advised. If you see all this iyalagbo, you see them, they'll put all those things, tie them and put them inside. When you come, they'll bring it out, it's dried. But some of them will still maintain their color. So when they want to deal with something like roots, those ones that are hard, most of them can be taken outside, because if it has high moisture, if you put them inside, it will start growing microbes. They'll start having some white things, meaning they are now contaminated. So those ones, you sun dry them. Another method you can do this drying is dehydration. Dehydration is safer than sun drying because in dehydration, you're using air, intense air, to take away the moisture without tampering with the metabolites. Okay, so at times you can also do freeze drying. You can imagine that whenever you're in a place like AC, you see with time your body will feel so dry, meaning the air from the AC is sucking in moisture from you. Okay, so in that same way, I can also use this AC to dry my plants. If I put plants under AC, the AC is going to take away the moisture from that plant and it's going to leave my plant dried without tampering with the metabolite. That's why I see some plants, when they're dry, they still maintain their green color or their original color because they're being dehydrated or they're being freeze dried. So now when I'm done drying these plants, I will then pulverize. Why do I pulverize? I grind or pulverize so that the absorption capacity will be higher, so I can extract more. So when I pulverize, then I'm going to extract. I can extract using different methods. I can use steam distillation. I can use distillation. When you're talking about distillation and steam distillation, these are used for plants that are not heat sensitive, or the metabolites you're looking for cannot be destroyed by heat. When your steam distillation is mild, more than normal distillation. In steam distillation, I'm going to use steam to distill, just like doing double boiler. While in distillation is normal, I'm heating directly. Okay, then we can also do pressing. In pressing, you don't heat, you use machine to press out. These are also done when you're dealing with something like seed. You want to take out oil, then you want to press it out. After drying the seed, then you have to use that machinery to take out the oil from that seed, because you don't want anything to happen to that oil. Remember, the oil obtained in this form is called carrier oil. It cannot be essential oil. Then we can also have what we call extraction. In some extraction method we are going to look at, we're going to look at what we call maceration, and other method we are going to talk about in other method of extraction. What is extraction? What do we understand by the term extraction? Without going into the details of grammar in extraction, extraction is that I have a leaf, like bitter leaf, and I want to take away the water, the bitter component of this bitter leaf. So I need to wash it. When I wash with water, I use that water to take out that bitterness that I am looking for. That saponin in that bitter leaf, the alkaloid in that bitter leaf, because I want to use it. What I need, I don't need the leaf. What I need is the water. So I need to take it out. So, but what is the water? Water is a solvent that we use to take out that bitter leaf and saponin from that plant. So this is a method, that is simple extraction. So that whenever you want to memorize the definition of extraction, you understand what is extraction. If you understand the meaning of extraction, you should be able to put it in your own words without changing the context. So you can say it's the treatment of plants, okay? It can be plant, it can be animal, with a solvent. In terms of bitter leaf, the solvent is water. Now, you can see some herbalists, when they want to extract, they're going to use alcohol. Some will tell you, put alcohol and water. Some they can call it, take 7-Up to soak it. These are solvents to take out the medicinal components of your plant or animal sample that you want to extract from. Okay, so this solvent is going to take out the medicinal active components from this your plant material or animal material, and the one that are not soluble, we call them inert matter. They become the residue you have. So after extracting, there's some things that will still remain. So the chaff will be there. That chaff is what we call the inert matter, which can never be dissolved, and we discard it. We call it mark. Okay. So, and what we have obtained from this extraction, the solvent is called the menstruum. Okay. We use solvents as menstruum. So my potential actives are stored in my menstruum while the mark is being discarded. So this is just the process of extraction in the grammar. But understand it that I'm using solvents to take out the medicinal components or constituents of plant material of choice. So just explain it so that somebody will know that you did not just cram, you understand what is extraction. Okay. Okay, we have different method of extraction. We have different method of extraction. The simple method of extraction we're going to be talking about is maceration. Okay, maceration is the number one method of extraction. Now, maceration is a cold method in the sense that it does not involve heat. Everything happens at room temperature. This is when I bring my sample, like I want to extract. Maceration is, it's not always advisable to macerate with water, except you're going to put it in fridge. Because maceration method is soaking my plants, my pulverized plant sample, while pulverizing because I want extraction to be harnessed. I want it to be effective. I'm increasing the surface area so this plant can easily penetrate into my plant material. So I pulverize, and I'm going to make it to be coarse. I will not make it to be too powdery. Too powdery is not going to help you, because at the end you're going to form something like putty that will be difficult for you to take out the liquid. So that's why we encourage for your sample to be coarsely powdered. In coarsely powdered, there is still pore sizes that can allow solvent to pass through. So whenever you want to increase your surface area, don't make it to be powdery, make it to be coarse. Okay. So when I put my coarse sample, I believe I am not in a haste. I can also use my sample unpulverized. I'll just soak my leaf like that. And remember, whenever you have your plant material like leaf now, well dried, when you use your hand to touch it, it will be breaking. So even if you don't want to coarsely do it, your hand can increase the surface area by visiting the plant material. Okay, this plant material, I'm going to soak it in a container. That container is advisable not to be plastic. It's always advisable, especially if you're doing a lab work, for you to use bottle. Why? Because there is a problem that the material in this plastic is going to interfere with the byproducts. Okay? And remember, not naturally, plastic is not healthy. So these plasticides are always in them. So it's wrong when you go and use all these plastic that are very light and soak your plant material. It's not healthy. Another thing that is not healthy that you see people do as a chemist, when you see them, you correct them, is when people go and use plastic to take agbo that is hot. You want to drink agbo, people that are cooking herbs on the road, you go and buy, they use plastic cup and fetch it for you. It is not healthy. And the worst of all is using that nylon to put warm agbo, nylon. Okay, you put it inside that transparent nylon, they put it and give you. It is not good. And when you use all these polyethylene bags, we call them polyethylene. When you use polyethylene bags and use them to put something that is hot, you want to buy akara, they use polyethylene bag to put hot akara for you. It is not healthy. It's always advisable for them to use paper to wrap it first before putting it into nylon, because these are the means that we are dealing with our system with plasticides. Okay, so we have to be conscious of how we use all these plastics, because virtually everything we use now is plastic. So when this material is being soaked, covered for a period of time, you can do 24 hours. You can also do 72 hours. That's what we'll talk about, 3 days. When you're soaking them, you're doing frequent agitation. You can be stirring, you can be shaking your vessel to make sure that a reaction or extraction is harnessed. After which, when your duration is expired, you filter. You can use cheesecloth, you can use filter paper. You can also use cotton wool to filter your extract. Remember, your menstruum is going to contain your extract, while the mark is the residue that you discard. So when you're done with the first extraction, it's advisable for you to soak that sample again with that solvent you use in extraction. You're just trying to rinse. You can see that the extra that is going to come out is also going to be colored, meaning that some of those extracts metabolites are still there. So you can agitate and filter again, maybe twice more before discarding the mark. So this method is what we call maceration method. Remember, I said it's a cold method. That's what we talk about. You allow it to stand at room temperature. So you don't heat it. You don't also put it inside fridge, except you're using only water as a solvent. Why? Because if water stay more than 24 hours, it's going to start having microbial attack. So your medicine becomes poison. And that is why it's always advisable for you to use aqueous ethanol, meaning you can add 30% to your 70% water. Let your water alcohol be 30% and your water be 70%. So with this, you can soak your sample for as far as 72 days. Okay, because there are some plants that can last without having microbial attack. Example of soft seed are cloves. Clove is something you can soak inside water because it has natural antibiotics. Okay, it's a natural antiseptic, so it's antifungal. So on itself, it can inhibit microbial attack. But whenever you're dealing with others, make sure either you add alcohol to increase the shelf life, or you put inside your fridge after 24 hours. That is a maceration method. Okay, because of our time, we're going to just talk about infusion and digestion. Okay, or maybe I think I should take your question. The next class, I will talk about other method of extraction. So let's use this 5 minutes to take question. So ask your questions. Ask your question. Now remember the norm. The norm is that I'm going to open a Google Classroom and we are going to have our classwork. So now I'm going to give you the question. Please, the course rep, take down the question for today's classwork. So I'm going to open the Google Classroom. I'll share the link. So you answer the question and send to the Google platform the way we did in 322. And remember, everything has limits. So as soon as we finish this class, you're expected to submit the assignment before 11:30. So take your question. Question number 1. Please, course rep, you're going to share this question to me as MS Word on the platform. I know that the platform was created. So you're going to share the question. Question number 1. What do you understand by natural products? If you've taken, tell me taking. Taking. Question 1. What do you understand by natural products? Adele Daniel, please take down the assignment. Question number 2. Question number 1A, what do you understand by natural products? Question number 1B, distinguish between types of natural products. Distinguish between types of natural products. In that question, you're expected to name the types of natural products, then distinguish them, okay? So when you're told distinguish between types of natural products, before you distinguish, you're supposed to name, list them, then distinguish. Number 3, mention 7 classes of natural products. That's C. Mention 7 classes of natural products, C. D of 1. Mention 5 classes of secondary metabolites. Mention 5 classes of secondary metabolites. Question 2. Show the biosynthetic pathway of synthesizing flavonoid and tannin. Biosynthetic pathway for obtaining or synthesizing tannin and flavonoid. That's number 2A. Number 2B, what do you understand by the term extraction? What do you understand by the term extraction? That is 2 words. That's where we're going to end today.
Please repeat question 2A.
No, it's the course rep that will repeat the question because I did not write the question anyway. Course rep, can you repeat the question? Daniel.
Which number?
Just start all over from 1 to 1.
Number 1A,
I think it's
what do you understand by natural products?
Okay. Continue.
Number 1B, distinguish
I think
types of natural products.
I think what you need to do is to go and share it on the platform. People that did not get it to get it from there. Okay? I think it makes sense so that I have a class now, so I need to end the class. So we're going to see next on Wednesday. I'm going to create a Google Classroom. I will share the link on the platform. So you do and submit on the Google Classroom. By 11:30, it will be late. And remember what it means to be late. I know you're used to it from 3 to 2. So I wish you the best. See you on Wednesday, the same venue, Zoom, until hostel is being sorted. We'll continue to do Zoom. All right. Thank you for coming. See you. Bye.